Okay, good morning. We're going to go ahead and get started here. We're still working our way through chemistry. And does anybody have any questions on anything going on right now? Okay, then I'm assuming everybody can hear me. And I'm going to go ahead and go into, it'd be nice if I'd open it up first. Let me go ahead and open up PowerPoints. And we are still in chemistry. And let's see. Yeah, looks like a good spot. Back to here. Okay. Now I think we are looking at the rate of chemical reactions. I think that was the last thing we got to see. Let me go ahead and put that on here. And if we've already done this, please somebody speak up and tell me, because it is Monday after all. So, okay. I think we're somewhere right around here. Okay. We were looking at chemical reactions. Our body is nothing but a mass of chemical reactions occurring all the time. And we are we're monitoring those chemical reactions, we're controlling them all. The advantage we have is that these chemical reactions work very well for us in very small concentrations. Because see, in chemistry, in a chemistry lab, you can change the rate of reaction very easily. Temperature, you can raise the temperature. Chemical reactions work faster at higher temperatures. If you're going to boil water, it works better if you turn the temperature up all the way as opposed to halfway. Water will boil faster. The rate of reaction occurs faster. If you increase the concentration makes reactions go faster. The more if some is good, more is better. If you're trying to make sweet tea and you add in 1 gram of sugar into a gallon of tea, it's not going to have much effect. You're not going to make very sweet tea. If you add 10 pounds of sugar, you're going to get really sweet tea. If you're doing baking soda and vinegar, which is probably a better example, you're doing baking soda and vinegar. If you want to have a really exciting reaction, add a lot of baking soda to a lot of vinegar at once, and you'll have a much more exciting reaction than just a little bit. If some in the lab, if some is good, more is usually better. The reaction rate goes faster the more reactants you add, and size matters. Only in this case, size matters smaller. The smaller the particle, the faster the reaction rate. See, particles move around. Particles are always moving around in solution, in the air. Air molecules are bouncing around. We don't see them, but they're bouncing around on us. If they bounce really fast, we get warm. That's the friction of them hitting our bodies. When you turn the heat up in the thermostat, you cause the air molecules, you transfer energy to the air molecules. They move faster. The friction of them hitting your skin makes you warmer. That's how it works. Small particles move around very faster than large particles. If you were going to make that sweet tea again, you would use a bag of sugar, the granulated sugar, instead of a block of sugar, because large particles don't move around quickly and don't cause a reaction to occur as quickly. So smaller is better when you're dealing with chemical reactions. Now, since our bodies are stuck at 98.6 or 37 degrees Celsius, we can't change the body temperature. Now, we know that during a low-grade fever, we see we see more efficiency in metabolic activity. So we go up 1 or 2 degrees, our cells become more efficient at killing bacteria. So we have a better reaction rate. That's the only time that we can really raise our temperature for anything to increase chemical reaction rates. Usually we're stuck at 98.6. We can't raise temperature just to get a reaction going. We can't do anything about the concentrations because we're already dealing with extremely tiny amounts in our bodies to begin with. And the particles are already small. So very, very tiny amounts. We can't do anything with it. The only thing we can do is use something called a catalyst. A catalyst makes the rate of a reaction go faster without dealing with particle sizes or temperatures or anything like that. A catalyst is a molecule, usually a protein, that we call an enzyme in our bodies that allow reactions to occur at a faster rate. All they do is they physically bring the reactants together. They bring these small particles together, allow the reaction to occur, and then release the product. And then they go do it again and again. The only thing we can do is we can produce large quantities of these enzymes, flood the area where a reaction is going to occur, and make the reaction go faster by simply bringing the particles together. Now, if we move on to the other aspect of chemistry, we've really been talking about physical properties so far. Physical properties involved in covalent bonding and diffusion and reaction rates. Now we'll switch gears a little bit and go into what we call the biochemistry aspect of chemistry in the human body. Now, chemicals are organized one of two ways. They're either inorganic, meaning they don't have carbon. They don't contain carbon is the atom that determines whether a molecule is organic or inorganic. Inorganic molecules don't have carbon. Carbon shows up in every molecule that's in a living organism. It's in the carbohydrates, it's in the lipids, it's in the proteins, it's in DNA. So everybody has carbon molecules in their body's chemistry. And that is what we call the organic side of chemistry, or the chemistry of life, if you will, or some call it carbon chemistry. The inorganic compounds don't have any carbon. It's water, it's all the salts, it's most of the acids and most of the bases. They don't have carbon. The only exceptions to this, that any inorganic compound that would contain carbon, is carbon dioxide and carbon monoxide. They're both gases, and they're usually produced as a product in either incomplete combustion, that's carbon monoxide, or carbon dioxide gets produced when we break down sugars, carbohydrates, for energy. Those are the only exceptions here, but they are not organic molecules. The organic compounds are the carbohydrates, the fats, the proteins, and the nucleic acids, the DNAs and the RNAs. They all contain carbon, they're all large, and they're all bound covalently. So they have lots and lots and lots of potential energy. We need both of these. Water is the largest inorganic compound in our bodies, making up about 70% of our body. When we're infants, it's closer to 80% or more. When we become older, when we move, elderly people have probably around 60% or even less. So that explains why they get wrinkles, because they lose body water over time, and so their skin becomes more wrinkled. It loses what we call turgor. When you've seen that when a plant is wilted, it doesn't, that won't stand up, and you add water to it, and it miraculously comes back. Turgor is what's missing in the skin of elderly people. The skin wrinkles simply because of their body water is lower. Water is an amazing compound. It is the universal solvent. It is what we dissolve everything into in our bodies. Water here. Water lines our lungs. We dissolve oxygen into water to get it to go across the cell membranes in the lungs to go in the blood. Water is what our blood cells float in. We call it plasma. And the plasma also contains the sugars and the hormones and antibodies and the salts that our bodies need. So it is the #1 transport medium in our body. Everything has to go into water. Even gases leaving the body, gases being carried in the plasma, for example, like CO2, is in water, and it has to pass through that water layer before it can become a gas. So we see water used for all sorts of things in our bodies. It dissolves all sorts of uses in our bodies. It's what we dissolve all the salts into, the sodium and the chloride, and the potassium and the chloride, and calcium carbonate and magnesium carbonate. All these salts, all these ions are dissolved in water. Now, water also helps us maintain our body temperature. That's the high heat capacity. We don't have a lot of swings in temperature because of water, because it takes a lot of energy to get water to a temperature to increase. So water's gonna absorb heat temperature changes so that we stay fairly constant. I mean, it's totally unrelated to humans, but consider if you've ever been to the beach in the early part of the summer, the ocean water is cold, hasn't warmed up yet. It takes all summer for that water to warm up. It's because the water has a very high capacity to absorb heat and not change. And so that takes a long, long time. By now, and in September, ocean water is probably in some places over 80 degrees. And it will stay warm for a very long time through the winter. It will still be reasonably comfortable in November because it won't be cold like it is in the spring, because it takes a long time to heat up, a long time to cool down. If you've ever been to the beach in the spring, you know that the ocean is cold. If you've been to the beach in August, you know that the ocean is very warm, and it'll stay warm for a long time. We use water in our bodies for cooling. We sweat, we perspire when necessary to give off, to release, to remove heat. Water evaporates, and when it evaporates, it releases lots of heat. So we use this for cooling. Okay, so those are the big three. There was another one, I think. Let me go back one. Oh yeah, cushioning. The water cushions our organs. Our brain floats inside our skull. Our brain floats inside our skull in a sac called the meninges. Our brain is not strong enough, physically strong enough to support its own weight. So it has to float. It has to float in a sac of salt water. We call that cerebrospinal fluid. So it protects the brain, it allows the brain to float, it cushions the brain. We have water in our fat cells that are acting as a cushion to protect lots of organs that don't have bones wrapped around them. Okay, now salts are really important to us. Sodium is the #1 ion in our bodies. It is the #1 ion outside of the cells. Potassium is the #1 ion inside the cells. And I'm not talking about the atom, I'm talking about the ion. Sodium ions and potassium ions. Remember, sodium ions are positively charged. They are cations, and so is potassium. These are called electrolytes. Salts make up electrolytes. Salts go in, a salt like sodium chloride, like table salt. If you pour table salt into a beaker of water, it will separate, it will dissociate into its appropriate ions. In this case, sodium and chlorine. Sodium is the cation and chlorine is the anion. That's what salts do. These salts are positively and negatively charged. They're attracted to each other, and they have a charge. We call them electrolytes. They give a charge to water. They can carry a current. Our bodies are charged. Muscle cells have a charge of -90 millivolts. Nerve cells have a charge of -70 millivolts. Electrolytes carry a current. The ability to contract muscles, the ability to generate and send nerve impulses are based on the presence of sodium ions and potassium ions in solution. So we depend on sodium, we depend on potassium, we depend on calcium, we even depend on iron. Iron is in the middle of the hemoglobin molecule and holds 4 oxygens per hemoglobin molecule. But it's the sodium and the potassium and the calcium. Chlorine doesn't really matter. Chlorine doesn't get to do anything. It's there, but chlorine is not a player in any of this. Chlorine doesn't get to play. It's just there because it's the other half of calcium chloride and potassium chloride and sodium chloride. It is not the important it's not one of the important electrolytes. Sodium is the #1 ion in solution outside the cells. It's in our plasma, it's in the interstitial fluid, it's in the lymphatic fluid. Potassium is the #1 ion inside the cells. So we have high sodium outside and high potassium inside, and that is very important to us. So these electrolytes are nearly always made up of ions, sodium and potassium in particular. And we see that what happens to an electrolyte when it goes, or a salt when it goes in the solution. Well, you've seen it when you add salt to water. The positive ends of the oxygen molecule are attracted to the negative ends of the salt crystal. Salts are crystals. And so the positive hydrogen end of a water molecule, it's a polar molecule, will grab the negative end of the salt crystal, the chlorine, and pull it away from the crystal. The negative end of the oxygen will grab the sodium and pull it away. And so all of a sudden you have this, all these salt crystals going into water, and the water just rips it apart, and salt will go into solution very, very quickly. Because ionic, salt crystals have positive and negative charges. They're attracted to each other electrostatically. It's a weak bond. and it's broken by the polar bond, by the polar molecule, the polar covalent water molecule with its positive and negative ends, grabs it and rips it apart. Now it doesn't destroy the salt. When salt goes into solution, it will stay there in solution. If the water evaporates, the salt's left behind. We don't lose it. Another substance that acts as an electrolyte in our bodies are acids and bases. An acid in solution will dissociate into its positive and negative components, just like a salt will. An acid is what we call a proton donor. Acids give up hydrogen ions. Proton donors. You know, what's a proton? Well, we know the proton is what's in the nucleus of an atom, the positively charged structure of the nucleus of an atom. If hydrogen, which has one proton and one electron, gives up its electron, become a hydrogen ion. You know, the only way a hydrogen can become an ion is to lose its electron. It's not going to gain an electron. It can't, because it only has one proton. But if it gives up that electron that balances the proton, if it gives up that electron, then what do you have left? You only have the proton. So the term proton and hydrogen ion are used interchangeably in here. So, example, hydrofluoric acid, HCl, will dissociate into the hydrogen ion and the chlorine ion. You know, to become an ion, you have to gain or lose an electron. Well, in this case, the chlorine has likely gained the electron from the hydrogen to make hydrofluoric acid. Now, when it goes into water, you have the hydrogen ion or the proton. You know, so acids are proton donors. They give off hydrogens. They get hydrogen ions. Hydrogen ions are the protons. Consider this. You've all heard of acid reflux. Maybe a number of us have taken medication for that. There are a lot of the acid reflux drugs are what we call proton pump inhibitors. In our stomach, we have pits, gastric pits, that release hydrofluoric acid. They are releasing protons. You know, they are pumping out hydrogen ions, making the stomach very acidic. These proton pump inhibitors block those pits so they can't generate these hydrogen ions. That's why they're called PPIs, or proton pump inhibitors. Protonics is a good example of a proton pump inhibitor. Prevacid, you've seen Prevacid. Prevacid is I'm sure you have anyway. You can go get it at Walmart. Prevacid is a proton pump inhibitor. You know, all of these drugs work by reducing stomach acid by preventing the action of the proton pump. Proton pumps pump out hydrogen ions to make hydrofluoric acid. Got to have hydrofluoric acid in our stomach to digest proteins. That's why it's there. Anyway, acids give off protons. The stronger the acid, the more protons they're going to release. Hydrochloric acid in our bodies, we use it all the time. Carbonic acid shows up in our plasma. It's what you get when you combine carbon dioxide and salt water. You get carbonic acid. We use that for a couple of things. We use it, it forms because it's carrying carbon dioxide. When carbon dioxide is released from a cell that just burned up a bunch of sugar, the carbon dioxide goes into solution in the plasma, forms carbonic acid. That's how we carry it to the lungs. And then we can release it at the lungs. The carbon dioxide also tells our brain, hey, guess what? We've got a lot of carbonic acid here, because we don't monitor carbon dioxide levels. we monitor acid levels. The more carbonic acid we form, it means as far as the brain is concerned, the more CO2 we have. And so if we generate a lot of CO2, we breathe faster to blow it off, simply because we formed a lot of carbonic acid. CO2 plus water is going to form carbonic acid. Okay, so hydrochloric acid and carbonic acid are the two big acids we deal with in our bodies. Now, if you're giving off protons, you have to have something to accept them. And those are the bases. Bases are proton acceptors. They grab hydrogen ions out of a solution. Sodium hydroxide. You've all heard of sodium hydroxide, I'm sure. It's the basis of oven cleaner, among other things. It will dissociate into the sodium ion and what we call the hydroxyl ion. The hydroxyl ion is the OH portion of a water molecule, one oxygen and one hydrogen, and they've gained an electron. So that hydroxyl ion is the negatively charged half of a base. So sodium hydroxide, classic example here of this. The bicarbonate ion, sodium bicarbonate, baking soda, you dissolve that in water and you'll have the sodium ion, which is the positive half of it, and you'll have the bicarbonate ion, which is the negative half of it. And so the bicarbonate ion, essentially no different than the hydroxyl ion, at least as far as charges go. Other base we have in our bodies is ammonia. We generate ammonia type wastes all the time. When we break down proteins, we generate ammonia. Proteins are made of amino acids that contain nitrogen, and ammonia is NH3. So bicarbonates and ammonia and hydroxyl ions are going to be in our bases. Now we monitor the concentration of acids and bases in our bodies using what's known as the pH scale. And the pH scale is simply a measure of the hydrogen ions in solution. That's all it is. How many hydrogen ions do you have in solution? The more hydrogen ions you have, the more acidic that solution is, with all the properties of a stronger and stronger acid. Now, the definition of pH is it is the negative logarithm of hydrogen ions in moles per liter that ranges from 0 to 14. Now, what does that mean? Because I will tell you, in all the years of teaching A&P, I've never had anybody run up to me and say, quick, what does pH mean? What you need to know on the pH scale is it ranges from 0 to 14. It measures acidity. Anything from 7 down to 0 is the acid side. Everything 7 and above to 14 is the basic side. Our bodies like to hang out right around 7.4. Our pH, the level of acidity in our bodies is right around 7.4. So we are actually slightly alkaline. It is a logarithmic scale, meaning every time it goes up or down 1 on the scale, it increases in intensity. So pH of 7 is, pH of 6 is 10 times more acidic than a pH of 7. pH of 7 is neutral. You're going down the scale, pH of 6 is 10 times more acidic than 7. pH of 5 is 10 times more acidic than 6 and 100 times more acidic than 7. So by the time you get down to pH of 1, you're very, very acidic. If you're going up the scale, pH of 8 is 10 times more basic than 7. 9 is 10 times more basic than 8 and 100 times more basic than 7. So you get more and more basic. Now, if you're getting numbers at the extreme ends of the scale, they're dangerous. They can cause tissue damage. They can hurt. The range is 0 to 6.99 is acidic, and 7.01 to 14 is basic. Neutral is pH of 7. The hydrogen ions and the hydroxyl ions are balanced. Distilled water is neutral, but the solutions in our bodies run at 7.4, pH of 7.4. We don't get excited until we go above 7.4. The range that we can operate in is between 7.45 and 7.35 pH. There's the scale right there. Human blood runs at pH of 7.4. It's already slightly alkaline, probably because CO2 is always forming in it to make carbon dioxide. The a few examples here. If you like coffee, coffee has a pH of 5. If you like orange juice or lemon juice, I don't know why you like lemon juice, it has or stomach acid, pH of 2. Hydrochloric acid has a pH of 1, or no, I'm sorry, pH of 0. Oven cleaner is a pH of almost 14. Pure sodium hydroxide has a pH of 14. Ammonia, pH of 11. Bleach, pH of 10. Egg whites are 8. So we see this. So why is this scale important to us? Because our bodies are designed to operate around a narrow range of pH between 7.45 and 7.35. If we go outside of that, bad things start happening. Cells start breaking down one way or the other. We even use this to measure how much carbon dioxide we have. The brain can't monitor every cell to look for CO2 levels, but what it can monitor is the acidity level in our plasma. As the pH starts dropping, if we go below 7.35 in our plasma, the brain will say, hey, we are getting more acidic. It must be because of carbon dioxide. So let's breathe faster and blow off that CO2 so we don't get so acidic. Why does CO2 affect our pH level? Because carbon dioxide, when it forms, has to be taken to the lungs. We have to get rid of it. The easiest way to carry it is to form carbonic acid. The CO2 combines with the water in the plasma, forming carbonic acid. The more carbonic acid we have, the more acidic our blood pH becomes. And the pH levels drop as the amount of acid goes up. So if the pH level starts dropping, the brain says, we got to get rid of this acid. Let's breathe faster, blow off the CO2. And our levels come back up. Now, acids and bases do something else for us. Acids and bases work as what we call a buffer. When you combine it, and I'll explain a buffer here in a second. Well, actually, buffers neutralize acids and bases. If you mix an acid and a base together, you will neutralize the acid and the base. If you mix baking soda and vinegar, vinegar contains acetic acid. Baking soda is sodium bicarbonate. Baking soda is the base, and vinegar is the acid. If you mix them together, you get a reaction. We've all seen that at some point. The volcano reaction that you get. You give off carbon dioxide, and you break apart the bicarbonate, sodium bicarbonate, and the hydrochloric acid, and you end up with sodium bicarbonate and acetic acid, and you end up with salt and water and CO2. Neutral. Unknown caller. When an acid and a base are busy neutralizing each other, you always end up with a salt and water, and lots of times a release of carbon dioxide, depending what it is. And that's what makes baking soda and vinegar so exciting, because you release a lot of carbon dioxide, but what's left over is salt and water. Now, we use these acids and bases in our bodies as a buffer. We don't like to get out of balance. We like to stay right at 7.4 in our pH. A buffer, if our pH numbers go up, if we go up above 7.45, we can start releasing hydrogen ions and bring us back down. An acid will bring us back down to 7.4 by adding more hydrogen ions. A base will bring us back up to 7.4 if we're getting more acidic by sucking up those hydrogen ions. And so we use acids and bases to keep our body, our plasma, our fluid in balance. So we can take a strong acid and convert it to a weak acid. We can convert hydrochloric acid into carbonic acid. We can, we will buffer the presence of strong acids and strong bases in our bodies. And the most important buffering system, you don't have to remember this again until AP2, is the carbonic acid bicarbonate method, because we use sodium bicarbonate in our bodies all the time. Sodium bicarbonate and carbonic acid work together to help regulate our blood pH at 7.4. Now, let's get into the more interesting side of this, the organic compounds. The organic molecules are anything that contains carbon except for CO2 or CO, which is carbon monoxide. Carbon works very well because it's a neutral atom and it binds very easily to 4 hydrogens. The most common covalent bond, other than water, is carbon and hydrogen. Carbon can accept 4 hydrogens. It will always share electrons. It never wants to give up an electron. It never wants to gain an electron. It won't. You'll never have a carbon atom. You'll never have a carbon ion. I mean, I guess you could, but it would be very, very unusual to have a carbon ion. It's just not going to work that way. Carbon doesn't gain electrons. It doesn't lose electrons. It always shares them. And it will nearly always combine with hydrogen, sometimes with oxygen, but it's hydrogen and oxygen it's going to combine to. Carbon shows up in all 4 of the organic compounds that human systems deal with. The carbohydrates, the starches, the breads, the pastas, whatever, the sugars, the lipids, which are the fats, the various types of fats and steroids, the proteins, and the nucleic acids, the DNA and the RNA. Organic compounds are polymers. They are long chains of carbon atoms linked together through covalent bonding. The nucleic acids are extremely long chains. They're classic examples of a polymer. A DNA molecule each molecule is about 6 feet long. Very, very, very long chain of carbon atoms linked together in a specific sequence. Polymers are made up of smaller units, so called monomers. The whole DNA chain is made up of subunits linked together. Fats are made up of smaller units of carbon linked together. Sugars are the same way. Proteins are the same way. Subunits linked together to make these long polymer chains. Carbohydrates are the classic example of a polymer chain. The carbohydrates include the starches, the sugars, as far as food goes, the pastas, the breads, the table sugar, sweeteners, rolls, anything, involving carbon and hydrogen and oxygen in a 1:2 to 1 ratio. Those are the carbohydrates. The purpose of a carbohydrate is to function as our body's fuel. Carbohydrates, important thing to remember, are always as it says there, the major source of cellular fuel, and that's in the form of glucose. Glucose is a simple sugar, and glucose is what our cells use for fuel. Our bodies will take a glucose molecule and oxidize it, burn it, and release the energy stored in the covalent bonds. Release the energy stored in the covalent bonds of the glucose molecule. Now, there are 3 types of carbohydrates: the monosaccharides, the simple sugars, the single sugars. These are the monomers of carbohydrates. The disaccharides are 2 sugars linked together. Most common disaccharide is what we use called table sugar, the stuff you buy at the store, in the 5-pound. Actually, no, they're in 4-pound bags now. Surprised, we were buying sugar over the weekend, and we usually buy a 25-pound bag at one time, and it lasts for 3 months or whatever. But it used to be sugar was in 5-pound bags. Now it's in 4-pound bags. So the disaccharides are the double sugars, the sucrose, the table sugar, the stuff that you get at the store, the stuff that comes in the white packets at the coffee bar, at Weigel's or Pilot's or any place you get your coffee. This is the stuff that we use all the time. The polysaccharides are the multiple sugars, large molecules of lots of simple sugars, single sugars linked together. The single sugar is the monomer. And so the polysaccharides are polymers, long chains of hundreds, maybe even thousands of simple sugars linked together. We use that to store energy in our bodies. Now, the monosaccharides, they look like this. Many of our monosaccharides have a ring structure. This is glucose that's spinning around on the screen there. It has 6 carbons. Those are the black atoms there. It has 12 hydrogens. Those are the silver atoms. And it has 6 oxygens. That's the red atoms. C6H12O6 is glucose. And this is the #1 simple sugar that our bodies use. Now, there are other sugars that we deal with. We have 5 carbon sugars. That's ribose and deoxyribose in RNA, ribonucleic acid, and DNA. Deoxyribose nucleic acid are genetic information, the nucleic acids. So we have simple sugars in 5 carbon. We have simple sugars in 6 carbon. Glucose, what we also call blood sugar or blood glucose, that we use to get energy to power our cells. We also have a sugar that's 6 carbon called fructose. Fructose is what shows up as a sweetener in most of our favorite soft drinks, Coke and Pepsi and Dr. Pepper and Mountain Dew. All these are going to be sweetened, unless they're the throwback version, they're all going to be sweetened with high fructose corn syrup. So that's the sweetener there. And then galactose, just a variation on glucose. They all look fairly similar. This is their chemical structure here. There's glucose, it's a ring. Most of these simple sugars come in a ring shape. Even the 5 carbon sugars come in a ring of 5. Here we see glucose on the left with 6 carbons. There's fructose in the middle with 6 carbons. It has 2 separate groups, one on either side of its 5-sided structure. And then the lactose on the end, which is very, very similar in shape to glucose. Just 2 minor changes in between glucose and galactose. The position of the hydrogens and the hydroxyl units on the ends of the molecule are flipped one to the other, and that's all. That's all that makes a difference. Now, these are the monomers that we build from. The disaccharides are the double sugars. See, glucose can go through a cell membrane. It's a large molecule, but it can go through a cell membrane with help. Facilitated diffusion allows glucose to get in. Insulin release, when you eat something and your blood sugar goes up, you eat a donut for breakfast or something, your blood sugar will go up because there's glucose now. Glucose levels in our blood have risen thanks to that donut. If so, then your body responds by releasing insulin. Insulin tells our cells to create more channel proteins or carrier proteins, tubes or diffusion units in the membrane that will allow glucose to come in. That's what insulin does. It opens up passageways for the glucose to enter the cell. See, glucose will fit, but sucrose, the double sugars, are too big to go through. Sucrose is 2 glucose. Well, it's two simple sugars. It is glucose and fructose together. And that is a huge molecule as far as the cell membrane is. So it can't go through. So we can't get sucrose into the cell. It has to be broken down to glucose and get glucose into the cell. So glucose and fructose give you sucrose. Glucose and galactose give you lactose or milk sugar. So you see, here we have, sucrose is glucose and fructose. Lactose, milk sugar, is glucose and glucose. And maltose is, maltose is 2 glucoses. Okay, the important one to remember, glucose and fructose give you sucrose. Glucose and fructose give you sucrose, which is what we eat as a table sugar. You can't taste the difference in any of these sugars. When you go to the store and you buy sucrose, you don't go, I'd like to get 5 pounds of sucrose. You get 5 pounds of sugar. And it says on the bag either pure cane sugar or pure sugar. The only difference between cane sugar and pure cane sugar and pure sugar is that pure cane sugar comes from sugar cane. Pure sugar comes from sugar beets. You can't taste the difference. They're identical. The only thing on the label that tells you differently is that you see one says cane and one doesn't. But when they're processed, and when the sugar beets or the sugar cane are ground up and we extract the sugar, tastes completely the same. I don't even know if there's a price difference on them. Anyway, the point is that we buy sucrose all the time, and sucrose is a molecule made from glucose and fructose. Now we see milk sugar in milk. We see malt sugar used in brewing, both in beer and in whiskey. Various flavoring additives there, and a source of fuel for the living cells. The yeast cells that are going to make our drinks, if you will. Now, the last category are the polysaccharides. The polysaccharides are made up of thousands of monosaccharides. In our bodies, we store our polysaccharides as something known as glycogen. Glycogen is a long chain of lots and lots and lots of glucose molecules. When we have excess glucose, we store it as glycogen in our liver and in our muscle cells. If we have even more glucose left over after that, we store it in our adipose tissue where it becomes fat. Now, plants don't store glycogen. They store starch. Lots and lots of glucose is linked together. Plants make glucose through photosynthesis, and they store that photosynthesis, the product of photosynthesis in the leaves and in the roots. You know, if you've eaten a potato, you've eaten stored starch. If you've eaten spinach, you've eaten the stored starch inside the spinach leaf or lettuce leaf or whatever. You don't get a lot out of the leaves. But the point is that most plants are going to store starch in two places, in their leaves and in their roots. If we eat the root vegetables, we eat stored starch. If we eat corn, we're eating the stored starch inside the kernels. If we eat an apple, we're eating the stored starch in inside the pulp of the apple. That's the stored starch of the plant. And we can convert that starch into glucose and then use what we need, and then the rest of it goes into glycogen. Now, the polysaccharides are not easily dissolved. They're not soluble. A lot of the polysaccharides are not dissolvable, and they form fiber, like the skin of an apple. We don't process that. It is fibrous. It's made out of lots of starch converted to cellulose. The same with the kernel of corn. If you eat corn on the cob, if you were of a mind to examine your fecal matter the next day, some people do, I don't know, I've been told this anyway, you would see the corn kernels in the feces because the yellow kernel or the white kernel doesn't get processed. We can't digest it. We eat the corn and we eat the contents out of the kernel. But the outer covering, the yellow portion or the white portion of the corn kernel made of cellulose, which is a polysaccharide, doesn't get processed. We can't digest it. So cellulose looks like this. It's very, very dense material. It's what lines the cell membrane, lines the cell. Plant cells have a cell wall, and cellulose makes the cell wall. We eat starch. All these are little glucose molecules linked together right here. We eat. We convert starch in our bodies to glycogen. This, very similar structure. Lots of glucose is linked together. We don't store starch, we store glycogen. And then cellulose is what we can eat, but it goes through our bodies as fiber, a high fiber food. If you like whole wheat bread, it's called whole wheat because the wheat kernel is surrounded by a layer of cellulose called bran. And whole wheat bread processes the whole kernel with the bran attached. The nutritious portion of the wheat kernel inside, I don't know what we call it, maybe the fruit, if you will. We use that, but the bran portion we can't process, so it passes through our bodies as fiber. And fiber tends to be a good thing, sort of a cleansing effect on our large intestine. Okay, I have to stop the class here. And we're going to cut it short today. I've got an appointment at 11 o'clock I have to go to, so I'm going to end us here, and we'll pick up with this on Wednesday. I hate to announce it like this, but this came up very suddenly, so I will stop this now. Anybody have any questions before I get us out of here? Okay, well, I'm going to end class. Enjoy your free half an hour. So I'll see you on